Hello, and welcome to the second part of eukaryotic features in our cell structure and function module. We'll go over peroxisomes and mitochondria, and then we'll touch on mitosis and some eukaryotic motility that we kind of touched on in the lab as well. So peroxisomes are named such because they contain an enzyme that can degrade hydrogen peroxide. Why would you want to degrade hydrogen peroxide? Because it's actually kind of lethal. When you put some peroxide on your skin, it readily turns into water and oxygen, and that um, carries the oxygen away and moves some of the debris and bacteria. But it also kills cells in the process, and so that is why we don't want it. Um, we don't want these reactive oxygen species, is what they call it. Peroxisomes also play a role in lipid biosynthesis, but we will see this in the lab, so you might want to know what the enzyme catalase does. Breaks down hydrogen peroxide, and it will bubble into water and oxygen. Well, the oxygen will bubble out of the water. We also have a cytoskeleton in eukaryotes that prokaryotes do not have. They don't need it as much. They have the inclusions to make them a bit stronger. But here we have a few features like actin and myosin that give our cell support. Okay, these are like bones for cells. Imagine if you put like a couple of sticks in balloons and you could assemble and reassemble them. It's also how some vesicles are transported, as you can see with the acetylcholine transport up there. And you can see that most of these filaments are made up of different types of protein. The second part of the cytoskeleton is the centrosome and centrioles. They're only involved during mitosis, during cell division, and they form a spindle that is used during mitosis. The spindle first aligns all the DNA to where it needs to be and then splits them in half. That is their function. The mitochondria will take center stage next week, and we will learn a lot about it, how it makes ATP. You may have heard growing up that it is the powerhouse of the cell. Well, it is, and why is it that, is what we're going to find out. It generates ATP, sure, but why is ATP energy? How do we get energy from ATP? The long and short of it, though, is that we need these structures, the mitochondrial matrix, and the cristae to set up a gradient that we are going to take advantage of. The cristae increases the surface area, and the mitochondrial matrix helps create that gradient. Chloroplasts are much like mitochondria, except they're photosynthetic. They create their own food. The thing to note about mitochondria and chloroplasts are they are the endosymbiont theory, where they probably came from, well, they surely came from two different bacterial species that was not degraded and provided a commensal relationship. So mitosis is the way eukaryotic cells divide. Again, we have to replicate our DNA. We have to replicate DNA, otherwise we won't have two copies, and one copy will only be there or will split us in half. So we replicate our DNA, we align ourselves up with our centrioles, and they create the mitotic spindle, and then they pull apart the bacterial copies, and then they will pinch in, creating two daughter cells. Okay. So we talked about membrane transport with bacteria, and we had active, facilitated, and active, passive, active, and facilitated diffusion and transport. In eukaryotes, we have endocytosis. and we can do so through phagocytosis, where you can engulf a cell. Pinocytosis is like cell drinking, where you can absorb nutrients from nearby cells, and you can also touch a receptor and cause a reaction. So phagocytosis is cell eating. It involves a engulfment of a large particles through the cell membrane. Um, some bacteria want to get engulfed, and they will take advantage of the fact that they're engulfed. Um, so they're hard to detect. Pinocytosis is just absorbing the nutrients from nearby, and receptor-mediated endocytosis, the requires a ligand or a special metal to bind to the surface. Okay, and then we have exocytosis, what we kind of mentioned briefly, where the vesicle um, can fuse outward and export kind of export contents without the phospholipid bilayer, and this is going to release secretory vesicles out of the cell rather than a vesicle transporting to another cell. Okay, and that is exocytosis. All right, and in the eukaryotic extracellular matrix, remember it is collagen, fibers, carbohydrates, protein, sugars, all of it, but it is not phospholipids. The phospholipids are in the phospholipid bilayer or the cell membrane layer. And we'll end our talk with eukaryotic motility. We saw these in the lab. Eukaryotic motility is pretty important. You did a whole lab report on it, and we want to see flagella, we want to see cilia. The cool thing about these guys is that they are exactly the same structurally. They have a 9 and 2 pair arrangement of filaments, and a cilia is just a short one, and a flagella is a long one. They both need to attach to a basal body and generate some kind of spin force in order to move. The cilia has numerous ones all around its body, like the paramecium pictures, and flagella could have one or it could have several. Okay, and with that, that ends our cell structure and function lectures. I am a cell biologist. It's one of my better subjects. So if you want to ask any questions on the cell, please do so, and let me know how you're doing.